You know, it's a real honor to be here today and give the keynote. Both Barack and Mark, the organizers, I want to thank you guys for making this happen. They both reached out to me to give me a little bit of guidance on what you should do to talk about a keynote, which I need, so that's good. And so Barack said, I really think your talk should be very forward-looking. You need to talk about the technologies that you're developing in the lab, to talk, specifically to address drug development and this thing called Eroom's law, which I'll talk about a little bit. And then a couple days later, Mark sent me an email and they said, in honor of DNA Day, I think this should be really retrospective. We should talk about sort of how technology has impacted the analysis of the genome. So I went to my wife and I said, what am I going to do? They told me two opposite things. Does that mean maybe I have carte blanche to talk about whatever I want? She said, I don't think that's a good idea. So we're going to try to get everybody covered, okay? So what I'm going to talk about is that I think an underappreciated concept in technology development is to allow your technology to do something poorly. Because if you can do that, it can often do something else, as Steve Jobs would say, insanely great, or at least well, okay? And that trade-off can be a very useful device to think about when you're developing technologies. Now you may say, this is obvious. Okay? Well, I can tell you for sure, it's not obvious to reviewers. Because when you submit a paper, the first thing they do is they say, your technology does this poorly. It's yeah, it was by design. Trust me. But also, when you're thinking about a technology, it's not something that comes to mind. You want to be a perfectionist. It's not something, first of all, many of the specifications that you think about in a technology are just understood by the field as being important. Long read lengths for Sanger sequencing, for example. So what I'm going to do is I'm going to talk a little bit about how I became aware of this technology when I was developing an early version of next generation sequencing technology in George Church's lab. And that'll cover some of the retrospective look-back analysis. And then I'll give an example, of another example of this concept being applied to a technology in my lab, and then talking about, the future. So in other words, if you believe in this concept, or even if you don't believe in this concept, where should you point your technology development brain cycles to figure out what the next thing to do is, okay? And so I'm going to talk a little bit about Eroom's law. We're going to time travel back to 1997-ish, and this is how DNA sequencing was done. This is how one version of it, this is actually the simplest version of how DNA sequencing was done. So you would take your genome, you would take your genomic DNA, and you would fragment it into lots of pieces, about 1 to 2 kb. You would ligate this into a plasmid. You would transform the plasmid into bacteria. The magic of bacterial transformation is such that each bacteria will get one plasmid, and then you can grow these bacteria up in wells so that you've now amplified. So you've cloned the plasmid, you've cloned your fragment, and you've amplified it. And then you had to grow lots and lots and lots of wells if you wanted to sequence a genome, because you needed tens of millions, hundreds of millions of fragments. So you would have a lot of these 384 well plates. You would handle all these samples individually by robots, and then you would do initially Sanger sequencing, and that turned to capillary electrophoresis, and that was how you did a genome. And one of the most important things in 1997 that people were working on was getting long reads. It was incredibly important to get long reads, because what you would do is you would get all these reads, and then you had to assemble a genome. You had to overlap them one with another. So if the reads weren't long, you wouldn't get good overlap. It just was a mess, okay? So a lot of the technology development, if they had a conference like this back in 1997, 50% of the talks would be about having long reads. So again, I was on a team in the Church lab there's George in 1997 where we were trying to develop a different way of doing sequencing, okay? And initially it was just George and I, and then we recruited Jason Dury and Greg Parecka. We were lucky enough to get them to sign up shortly thereafter. And it wasn't just us, there were other groups working on next generation sequencing, of which we were blissfully unaware of their efforts. So at that time. So actually, initially, we weren't thinking about sequencing at the very, very beginning, okay? What we were trying to do was actually make microarrays, because microarrays were all the rage, and we kind of wanted to get in on that fun, okay? And the idea was we would take a cDNA library, okay? And we would then put universal primers on either side of the cDNA library, and we would pour a acrylamide gel so that this library had very dilute, it was a very dilute, the gel had a very dilute amount of DNA, so these molecules were well separated. And then we would do PCR on this acrylamide gel so that each single molecule would amplify, but its products would remain very close to the original products, and you would get a colony of DNA. And we called them PCR colonies or polonies, okay? And then the idea was, once you had one of these gels, you could actually replica plate these. So you could just stamp another acrylamide gel, transfer DNA, and then do a PCR on that one, and then do another one, and do another one, and do another one, and mass produce it. Okay, that was the original idea. And in fact, you can see here that this actually worked. This is the original gel where we had a bunch of single molecule like cDNAs that were here, and then we just replica plated onto this one, okay? But the problem, of course, is now you gotta sequence one of these arrays so that you know if you've mass produced thousands of these. We never did that, but if you've mass produced thousands of these, you would now have to sequence one of them. And so we were thinking about different ways. We had a bunch of ways kind of lined up, and then what we sort of were like, well, first of all, let's figure out how many bases you actually need to uniquely identify an RNA so you can identify all the spots. And it turned out to be remarkably few. So in yeast, you only need 10 base pairs to uniquely identify about 2/3 of the genome, and at 11, you start to saturate. You're doing great. You can basically do this, okay? So then we thought, well, wait, what's the point here of doing all this work with the replica plating? Let's just use this for sequencing. And so this is the scheme we came up with where we would just, this is sort of familiar now with next generation sequencing. We would do sequencing by synthesis. We'd add a fluorescent base. We would cleave the base, we would scan the gel. We would see where it was incorporated. We'd cleave the base, add the next base, et cetera, okay? And so this was kind of interesting because this is where we had a clash of the kind of current culture. We thought, oh my gosh, we can get away with just a very, very short read lengths and even do sequencing. And when we talked to other technologists in the field, it was very muted response. We're not really interested in read lengths. I mean, they're like, I spent all my time trying to push this from 1,200 bases to 1,500 bases, and you're now talking to me about it. Don't talk to me about a 8 base pair or 9 base pair or 10 base pair sequencing technology. That's nonsense. And it was actually hard for us, too, to really internalize it. So when we published our first paper, we sort of figured this out actually like pretty quickly, like a few months into the project, maybe 1 or 2 months in, we had done this analysis and kind of committed to sequencing. But we kept the replica plating as a backup plan because we just didn't, in our hearts, really believe that we could allow our technology to do something poorly. Okay, so when we published our first paper, actually, again, it had the replica plating as a backup. But importantly, what this did is we also were able to show that you could get these PCR colonies, and you could get a large number of colonies. And then, sort of a couple years later, we were actually able to sequence a small number of bases. And these were wimpy reads, only 8 base pairs that we could sequence. So you can just see here, yellow means it incorporated. So for example, in this particular spot, it didn't incorporate here, it incorporated here, here, and here. And so the sequence was AGT. So we got maybe, there were maybe tens to low hundreds of polonies on the slide. We could get 8 base pair reads, but at this point, we were drinking the Kool-Aid. We were very happy about this. And we thought this was great because it showed that you could integrate the cloning, the amplification, and sequencing. And so all in one format, just with the handling of one slide. And this meant that you could then, if we could just scale this up and get slightly longer read lengths, we'd be home free. Well, it turned out scaling up was actually really, really hard. It was not so easy. So Greg, Parekha, and Jay really led the charge here. And we changed a ton of things on the protocol. And so we finally got sort of on the order of tens of millions of features on a slide and pushed the read length up to 13 base pairs now. So we can now do 13 base pairs instead of 8. But the other thing that Greg and Jay really developed that was instrumental is they figured out a protocol to do paired reads to get mate pairs. So it was 2 wimpy 13 base pair reads, but they were separated in the genome by about 1 kilobase of distance away. And this allowed you, this actually was incredibly powerful. It was way more powerful than just getting a 26 base pair read, because what happened is you could deal with repetitive DNA and you could have 1 13 base pair. If you had 1 13 base pair in a repeat, it would map all over the genome, but the other 13 base pair would map outside, and you could immediately lock in on where that was. And so, we sequenced an E. coli genome to high accuracy, so less than 1 in 10 to the minus 6th consensus error rate, and the price was about 1/10 of the cost if you were to do it by Sanger sequencing. And so, importantly, the 454 team, Margiles et al., and Jonathan Rothberg and Mike Weiner, also sequenced the mycoplasma genome, really like within 2 weeks. These 2 genomes were published, and these were the first 2 published genomes sequenced by next-generation sequencing technologies. So these crappy short reads actually worked out pretty well, and this was sort of, I think, very gratifying to us, and I think it was, this sort of was the turning point where, you could get away with these short reads and people saw the value in it. And we weren't the only people working on this technology, so at this point there were about 5 groups here. They started 4 different companies. They either collaborated with companies. We partnered with Agincourt, Solexa, which was Balasubramanian and Klennerman, and Kawashima and Mayer kind of combined their efforts and then put out the Solexa machine, which then ended up being bought by Illumina, and this was sort of the most robust. So all of these platforms worked. They all produced cost-effective bases, but this ended up being the most versatile and the most robust platform, and Illumina bought Solexa, and then this became the dominant platform. Most of the bases that were sequenced by second-generation sequencing were sequenced by the grandsons of this machine here. But what was interesting and very kind of, I thought very interesting relative to this story is in about 2006, the NHGRI had a program for next generation sequencing program, and a lot of these groups got together just like we are here, and we would compare notes. And one of the big talking points was that other groups had exactly the same experience, where they were just like, people would just be like, 20 base pair reads, that's ridiculous, right? And so the commercialization was a really important step in this thing. It brought a lot of capital, a lot of talent, and that capital and talent allowed a drop in the cost of DNA sequencing, so an exponential decrease year over year. Okay, so every year, the cost of DNA sequencing from 2007 to 2022, every year the cost of DNA sequencing dropped by a factor of 2 on average. Okay, so this is like Moore's law. You may have heard of Moore's law, which is the reduction of the cost of a computer reduces by half every 18 months. This is faster than Moore's law, okay? And this is still far shorter, even today. Here we are now, it's 20 years later. The reads are still far shorter than the state-of-the-art Sanger read length in 1997. So again, if you think back to this, what the trade-off was is short read lengths, meaning you could integrate all this cloning, amplification, sequencing, and this means it was possible to scale. And this scaling led to essentially a decrease of the cost of sequencing genome from $100 million to $400. $100 million to $400 over the course of about 20 years. All right, and so now if we're going to look retrospectively, how has technology impacted the analysis of the genome? I think the answer to that question, to Mark's question, is really there was a 100,000-fold reduction in cost of genome analysis. So we had one genome which was remarkable. Now, one caveat is that once you've got the first genome where you have to assemble it de novo, it's much, much, much easier to resequence a genome. That's a big caveat. But now it's estimated there are over 2 million genomes, and greater than 30 million exons have been sequenced, and this is part of the standard of care. And I think not only that, Jay and Erez had a really nice review article where they said that next generation sequencing has sort of become a microscope. You just hook it up at the end of all your favorite biochemical assays that existed, and then now it's become a microscope to study cellular function. And we've seen some superb examples of that so far today. Okay, so now we're gonna just say one other quick comment about this. So, you're saying, Rob, okay, this is the big trade-off that second generation sequencing made was read lengths versus scalability, but what about all this cool third generation sequencing that I'm hearing from Winston and Andrew and everybody here? How does that fit into the thing? Doesn't that actually kind of kill your thesis? And I would argue, actually, no, it's just something different. So in the early days, in particular, when you look at like Winston's talk, the early days, the trade-off was raw sequencing accuracy. In the early days, the raw sequencing accuracy was very poor, and it was now the second generation sequencers' turn to be the mean reviewers saying, it's never gonna work because the raw sequence accuracy is 80% or 85% or 90, whatever it is, right? And yet that trade-off means that you can do amazing things and get incredible long read lengths, and I think the technology is extremely exciting. So I do think it's kind of, I think it's kind of an interesting concept, and so we thought about this, so now I'm gonna transition to some of the things that we're working on either now or in the recent past. We're very interested in gene regulation. Transcriptional gene regulation is sort of central to almost everything that cells do. Gene expression is controlled by transcription factor binding, so measuring where a transcription factor binds is of central importance. And most technologies that measure transcription factor binding use antibodies to do that, and they're great because they avoid the need for genome engineering, which is super laborious, and they avoid the need for transcription factor overexpression, which is artificial, it's not biological. But if you do say, hey, I'm gonna take the hit on this, I'm gonna let my technology do this poorly, one thing you can do is you can actually now you can engineer a transcription factor to deposit a piece of DNA where it binds. And then you can engineer this piece of DNA to do different things. So for example, we've engineered this piece of DNA to transcribe into the genome so it actually reports its location in the RNA fraction of the cell, okay? And that's actually super useful. So one thing you can do is since it's reporting its location, so we use transposons, that's what we're using here. So this piece of DNA reports its location in the RNA, and so we call this a self-reporting transposon. And so what we can do then is we can just take the cells, and using standard single-cell RNA sequencing technology, you can simultaneously map transcription factor binding and mRNA expression at the same time. So that's one thing you can do that's neat. And this works. This is just sort of examples from single cells. So these are kind of binding peaks. This is the ChIP-seq data, bulk ChIP-seq data that's showing that it works. We can recover motifs. And then, of course, because you get the RNA fraction in the same cell, you can do this in complex tissues like the mouse brain. And you can basically say, where does the transcription factor bind in this cell type versus this cell type? Okay. But the other thing that's really neat, and this is a collaboration with Sid Perham, who you'll hear more about later, and Riley Mullins, a very talented grad student. You can just put a barcode in because you're putting a piece of DNA, you can just barcode the DNA, and now you can do multiplexed transcription factor binding in one reaction, okay? And so we can do 10 to 20 transcription factors at once. In this experiment, Riley did 8. These are all transcription factors that have the same DNA binding domain. These are the different binding patterns. So this is the clustering of the binding. And what you can see, it's super interesting, is that these are synthetic TFs that have the same DNA binding domain, but they have very different, like for example, very different binding preferences. Here's a region, here's a set of genes that are bound by this transcription factor, but are not bound by any of the other transcription factors, okay? So we're extremely excited about figuring all this out. We think this is going to be a rich source of transcription factor biology and biophysics to dissect. Okay, and then the other cool thing is that because this is you're inserting DNA into the genome, it's a permanent mark that survives cell division and cell differentiation, so it's a permanent record of binding. So what you can do is you can record transcription factor binding in a mouse brain, for example, okay? You can induce seizures in the mouse, and you can divide the mice into two categories, mice that had mild seizures and mice that had severe seizures, okay? And then you can harvest these transposons and look back in time and see what the enhancer usage in the brain was before they were exposed to this drug, okay? And so what that allows you to do is it allows you to correlate enhancer usage pre-stimulus with the phenotypic outcome, okay? And so this is a bioRxiv paper. This is a collaboration with Tim Miller and Joe Doherty, their labs. This is led by Ben and Mary. So you can check out their preprint. So Eroom's law, we talked about Moore's law, right? This exponential decrease in the price of transistors is named after Gordon Moore. Eroom's law is Moore's law spelled backwards, right? Eroom, Moore, right? Okay, I didn't come up with it. And the reason why it's Moore's law spelled backwards is because the cost of making a drug is increasing exponentially. So it's doubling once every 9 years. Okay, so why do I think this is arguably the most important question in biotechnology? Well, right now it's still profitable to make a drug. So people are still investing. Companies like Pfizer, companies like Merck are still investing in making a drug. But we have an exponential decline. Luckily, it's slow. It's once every, a halving every 9 years. But once this becomes no longer profitable, the amount of investment in our field, in biotechnology, will plummet, okay? Both because the drug companies won't invest in it and because, the NIH is the National Institute of Health. We have other institutes that have much smaller budgets that are more focused on, sort of specialized academic questions. But if this equation doesn't make sense, I'm skeptical that the public's going to want to fund it at the levels that we've been at. I think maybe, yeah, anyway, I'm not going to comment. So it used to cost $16 million to bring a drug to market in 1950, and now it costs $3.5 billion. Okay? It's interesting when I talk to people in academia about it, either one, they haven't heard of it, which I think all of you kind of fit in that category. Or two, they say, well, isn't that something for the pharmaceutical companies? Like, that's their business, right? That's their shtick. So I think I have a couple answers for that. So one is that, there's a great article that just came out by Gardner and Kinch in Stat just this week. Academia made IP contributions to 50% of all FDA-approved drugs from 2020 to 2024. So it's not just industry shtick. We do this, and we contribute a lot to this, right? So it is our problem. Second thing is that I think, again, where is it better? I mean, risky ideas are best incubated in academia, I think. I think this crowd would agree with that. For example, nanopore sequencing is a crazy idea from I don't know, it was in the 90s, so it's 30 years ago now. Crazy idea. It would not have it's super cool, right? It's the coolest thing. It would not have seen the light of day if not for work at academia. So we need new technologies to develop Eroom's law, and the new ideas are going to come from academia, I think. It's not to say industry isn't great. Industry has an incredible role, but for the really high-risk stuff, it's going to happen here. And again, I said, this is why I think this is a critical problem for the industry, because of the exponential decrease. So this is what a lot of my lab is now focused on, and what a lot of the MGI is focused on, is addressing this issue. I'm going to give you an example. So one is I think we need new technology. So, I think if there's one take home that I want to give to you all that are developing technologies, think about this problem, right? 95% of you did not raise your hand, so you didn't even know it was a problem. At least think about it and maybe you can even think about the whole allow something to do poorly trick. But the second thing is that, even taking existing technologies and just directing it towards, like, being aware that we should be trying to develop new drugs actually can pay dividends. And that's what I'm going to tell you about now. So this is a close collaboration with Sid Perham, who's a really good friend and collaborator. So Sid is an oncologist. He works on head and neck cancer. He's a surgeon. He cuts out head and neck cancer, and he's a fantastic scientist. And this is the team, Ananya and Fudong, and then Zhang Tai was originally on the team but left. So Sid, when he did his postdoc, he was studying head and neck cancer. He did a single cell sequencing study of oral squamous cell carcinoma, and oral squamous cell carcinoma, tobacco. So it's really tobacco is a huge risk factor. It has very poor outcomes, and the poor outcomes are really driven by metastasis. So cells invade. So you have a primary tumor. They invade through the ECM. They get into the bloodstream, and then they metastasize. So the oral squamous cell carcinoma tumors are really invasive relative to other cancers. So his question was, why are they so invasive? And what he found is that there was a gene program. He found a transcriptional program that is largely localized at the edge of tumors here that causes invasion, okay? And he called this program PEMT. And this program, I think, is notable for 2 reasons, okay? So 3 reasons. One is it regulates tumor cell invasion, but 2 is that it is an incredibly predictive biomarker. So it predicts outcome. The presence of this transcriptional program in a tumor predicts outcome better than smoking history in these patients, which is remarkable because this is really sort of a disease of smoking, okay? Second thing is that it is driven by a single transcription factor. So PEMT is driven by a single transcription factor, SNAIL2, okay? And so if you knock down SNAIL2, you can then do it. This is like a tail vein injection. These are mouse lungs. And what you can see here is that if you knock down SNAIL2, there's no metastasis to the lungs, okay? But when you have just the normal tumor, you see substantial metastasis to the lung. And he did a number of other studies that showed this, okay? So the problem is, so it would be great to therapeutically target PEMT. It's never been targeted. This program's never been targeted. It seems to be really important for outcomes. The problem is that SNAIL2 is not druggable, okay? So again, we're gonna kind of do standard things with the technology. So I talked to you about the transposon mapping technology, the calling card technology. But what we did is we used calling cards, the transposon technology, to map SNAIL2 binding. And we found all the direct targets of SNAIL2. And our hypothesis was that the direct targets of SNAIL2 ought to be druggable, okay? They should be therapeutic candidates. And so the way we test this is we just mapped, we identified about 100 targets by mapping the binding and then by doing RNA-seq and the SNAIL2 knockdown and then the wild type. And then we measured invasion in vitro. So this is what an invasion assay looks like. You just take the cells, you put them in a chamber that looks like this. There's a Mitra right here, and the cells invade through the Mitra if they're invasive, okay? And what you can do is you can just stay in the cells, and you see this is sort of like where the cells, so the cells were all on the other side of this, and they've just kind of invaded through the Mitra, and now you stay in them, and you can see, like, if you see a lot of purple, that means they're invasive, and if you see a lot of white, that means they're not invasive. So we took all the targets of SNAIL2, and this was a Herculean effort by Ananya and Fudong. They made 132 CRISPR lines, and what they did is they knocked down all of the targets of SNAIL2. So these are all different targets of SNAIL2. And what was actually, I think it was quite remarkable, is 66% of the genes that we tested showed a phenotype in invasion. So this is, for example, the non-targeting guide, and then you can see that all of these genes show significant decrease in invasion. It's kind of remarkable because I don't understand why so many genes are necessary for this phenotype, right? I figured it'd be a couple genes that would be important. SNAIL2 would regulate other things, but they're all necessary. Okay. But what we did is, so this is kind of, again, pretty standard stuff, but I guess the thing that, I might not have done before I learned about Eroom's law is we looked at all of our hits and we said, which of these have small molecule inhibitors? So it turns out that 11 of our hits have small molecule inhibitors for other indications, heart disease, diabetes, et cetera. And so then we took the small molecules and we found that 9 of the small molecules that we tried, or 9 of the targets had small molecules that reduced invasion in our in vitro assay. And so you can see here, for example. And so the thing that's really neat about this is that none of these small molecules are toxic to either normal cells or to tumor cells, okay? So they have almost no effect. So the IC50 is like, for example, 46 nanomolar here, meaning 46 nanomolar, you're only killing half the cells, but these things are efficacious. You can get 10-fold reduction in invasion even when there's only 2.5 nanomolar of drug being used, okay? So we think these are gonna be safe. Now you'd have to use them in conjunction with some other treatment, either surgery or radiation or some standard, chemotherapeutic. But we think this will be really a safe drug, which is, sort of unique for this type of thing. So then we decided to do some in vivo assays. What we did is we just did a tail vein injection. So we would take cancer cells and inject it into a mouse tail brain. Meanwhile, we're pre-treating the mice with these drugs, and then we're asking how much metastasis do we see in the mouse lungs, and we'd read this out by a luciferase assay. And so what I hope you can see here is that in the control mice, there's a lot of metastasis to the lungs here, because you can see a lot of luciferase signal in the lungs. However, with this drug, 10 milligrams per kilogram of this drug, which is within the therapeutic range of a dose, you can see there's almost no metastasis to the lungs, okay? And so this is just a quantification of that. So this is one hit. This is one of our hits, but of our 9 that had activity in vitro, we tried 5 in vivo, and 5 of the 5 that we tried worked. So we think we have 9 novel therapeutic candidates, and they're all sort of different genes. They're not 9, of the same molecule targeting the same gene. So we're pretty excited about that, but I think that, again, this is something we might not, actually Sid would have tried, but I would not have thought about necessarily if I had not been aware of Eroom's law. Okay, so I think I've said most of this. This pathway has never been targeted, so we're excited about it. It should be safe because it's not toxic. And we think these therapeutics might be useful in combination with sort of standard anti-proliferative therapies. All right, so finally, the take-homes. Hopefully I've convinced you that allowing your technology to do something poorly can be useful because sometimes that allows you to do something else insanely great. Certainly, I think it almost goes without saying that technology has had a large impact in the study of genomes and science projects, and I think this crowd is obviously preaching to the choir. Eroom's law is a major existing challenge for biomedical sciences. I think we have to beat it or we'll see dramatic decreases in funding, and I think that this challenge is ripe to be overcome by technologists such as yourselves. So that's it, and I think I got everybody on the way in terms of acknowledgement. So I'll take any questions. Thank you.
All right, wonderful. Thanks, Rob. Start over here with Winston.
I love the talk. I love the idea that things have to start bad. But here's my question. This is a special sort of safe place, right, where tech dev is allowed or encouraged.
Yeah.
But often if you submit to general study sections at NIH, you get this whole phenomenon that either you're not threading the needle that this will never work or this has already been done, right, is what I often see at general study sections. And I'm not trying to have any political comments, but let's face the reality that we're probably looking at a big shift in how science is funded in the US.
Yeah.
And we can't go back necessarily to the Bell Labs model where you used to be able to screw around too, which is how we got silicon microfabrication and electronics. So how do we convince pharma that academia maybe is the place to fund risky research? Or how do you see, with your sort of experience and from the position that you're sitting in, how you get this kind of work that I agree is critical, and I'd never heard it articulated as Eroom's law, but like I was aware of the problem. How do you see a path forward to continuing to do tech development, to convincing people that we need to start with crappy methods that have potential? They have the spirit. How do we convince them of this?
Well, that's a great question. I think, to be honest, the first thing to do is to talk about it. I mean, I think I have to say, oh God, Winston, I got to confess here. When I got a fair number of like nanopore papers and
Sure.
and PacBio papers in the early day, and I would start to read it and I'd be like, these guys have error rates are 85%. I'd do the calculation. I'd be like, and I'd be like, this is, and then I'm like, what am I doing?
And you know what I would always say? Does it map? If it maps, we're done. You know
exactly. And then it's like, and there's so many ways to, right? I mean, obviously,
Sure.
there's so many ways to get around it. But I mean, you just go to that place in your head because you're putting on your critique hat, right? And so
science advances one funeral at a time.
Yeah, exactly. And so when you kind of acknowledge this, right, that this is an important feature of technology development, I think it helps because then people are going to say, first of all, I think it helps. One is in part, to all the current and future reviewers, keep this in mind, so be nice. And then the second thing is that I think when, you're talking to somebody in pharma, I think they recognize, like, man, I've got Excel spreadsheets that tell me what my bottom line is, and my manager says this and this and this. And so I think that this is appreciated. I think it's just, we just need to kind of, give examples. I mean, I think, like for example, nanopore sequencing is a good example. Next generation sequencing is a good example where they were really crappy in the beginning. And I think just beating people over the head with it is one way to start.
Yeah.
I don't have any solutions to the kind of the global problems, right?
But I mean, I think, I'll just say, I think convincing people that we're cheap to throw a little bit of money at to do something risky, as opposed to hiring like an FTE at a company or things like that, because we'll try anything. That's why we like this stuff. You know, we like living on the edge and pushing what we can, seeing what we can do. So I think that there's room for innovation, but I think we got to be able to think nimbly as things are shifting under our feet.
Yeah, I also think just one last thing is it's super important, and you know, the review, I really encourage everyone to read the stat article by Kinch and Gardner. Yeah. Articulating where we make a contribution better is also going to serve us well, I think. So.
So the cost of building a house in California probably follows Eroom's law. Maybe not with the same exponential decay rate, but it follows Eroom's law. And I'm just wondering, and it's not because of a lack of technology. What's that?
Come to Missouri.
Yeah. It's not because of a lack of technology in building houses, right? It's because of increasing regulation. And so I'm just wondering if how do you parse out Eroom's law between like this increased regulation?
Yeah, it's a great question. I'll be honest. So there's a lot of articles about Eroom's law. So I have one citation of the one that I put in there, but there's probably like, I think Mike Kinch is actually a big author that has done this. They've listed like 6 reasons why Eroom's law is there. Regulation is one of them. Over-regulation is one of the reasons. There's the better than the Beatles problem. There's a lot. I'll be honest, I'm flummoxed. I don't understand, like empirically, I understand that Eroom's law is there. I don't understand what the root cause of Eroom's law is. So, you know, I highly recommend everybody reads the reviews on Eroom's law. And, but I go through the list and I'm like, yeah, that's a factor. Yeah, that's a factor. But you know, the thing that really bothers me is it's an exponential increase in cost. It's not linear, it's not polynomial, it's an exponential increase in cost, despite the fact that we have an exponential increase in the number of bases you can sequence per dollar, and an exponential increase in the number of computational cycles you can perform per dollar. So in the light of those incredibly important technologies, we still have an exponential decrease. It's really hard for me to explain by regulation or by, right, those things. I don't know. You know, the one that kind of resonates with me the most, perhaps, is that, you know, we've gotten all the low-hanging fruit, right? In other words, like the druggable molecules that are out there, we've identified, we've targeted them. You know, the molecules that can be given systemically and not in a specific cell type. You know, like there's targets where it's like, I know this protein would cure disease, but I can't knock it down in some other cell type, otherwise it's going to cause another disease, right? So, you know, so I think that's part of it, is that we've got, and I think that's why I think we need technology. Like, for example, you know, can we figure out how to deliver nucleic acid therapeutics, right, in a more effective way? That's a technology problem. Then that just opens up a whole new class of proteins that can't be targeted by small molecules. Can we figure out how to get small molecules to target one particular cell type and not the rest of the cells in your body, right, in a reliable way, in a very modular way? I think those are like incredibly important questions, and that would get over this low-hanging fruit problem. I think that's the one that resonates the most with me, but none of them really resonate with me that much.
Thanks.
Come on.
So I was just kind of wondering from your unique perspective, not that this is bad in any ways, but all of our current sequencing technologies, except nanopore and maybe sequencing by ligation, are sequencing by synthesis. And I think it makes sense to continue optimizing these, what we have so far. But I was just wondering if you or anyone else, or maybe even a fever dream you woke up from, anyone else pursuing another angle that is truly a new type of chemistry like the nanopore did at this time.
You mean, so sorry, what your question is are there other sequencing chemistries other than, that aren't sequencing by synthesis?
More just that we always take advantage of the base pairing to do sequencing. And I'm not saying that's a bad thing, but
yeah.
you know, now we had the nanopore that came at it from a truly unique direction. And I'm not saying that's what we need to do. I was just curious if you've heard or thought of any other variants of this for sequencing that do take advantage of a truly unique aspect.
Yeah, I mean, I think in the old days there were an enormous number of cool ideas that were out there. There was sequencing by hybridization, that's still sort of interesting. There was sequencing by degradation, where you would sort of degrade DNA and then send it through a nanopore typically. And there's sort of a number of others that are out there. I think what's happened though is that you have to think about the total cost of a protocol, right? So once you get down to where sequencing is less than some number, then your sequencing isn't your rate limiting step when you're doing an experiment. It becomes just right? Like sequencing, for example, for a whole genome when it was super expensive was super rate limiting. If your sequencing cost is less than the cost of getting your blood sample from a patient and then doing the DNA extraction, then the value of taking the sequencing cost from $100 to $10 is not really the bottleneck, right? So I feel like to a large extent, a lot of the diversity and sequencing ideas have been kind of wiped out for two reasons. One is that it's gotten really, really cheap, and now we need to think about how are we going to get the same level of analysis for proteins? How are we going to get the same level of analysis for modifications to RNA or to DNA, right? And so I think there's still a lot of innovation that needs to be done there. And I think that's where there's going to be a lot of really, we've heard some already, right, here. So I think that's where a lot of people are now thinking. So I think there was a lot of diversity, and that kind of got swept out because sequencing by synthesis really started working. And once you get it beyond a certain point, people are just like, I don't really need to, this is no longer the rate limiting step. Does that make sense?
Yes, very. Thank you.
I think one of the technology areas that's grown by leaps and bounds in the last handful of years is spatial omics technologies. It seems like an area now that is, there's made a lot of progress starting from a poor technology starting place, perhaps, especially when we look back. What do you see as the challenges and opportunities there?
So I think that's a great question. I think that so from a purely technology point of view, I think one challenge is that you have these hybridization-based technologies, like sort of single molecule, or I say single molecule, but it might have a rolling circle amplification step. Single molecule-based, probe-based, which are really highly sensitive. In other words, if you have a transcription factor that has 3 RNA copies per cell, you can still detect it. And then the problem is that you're limited in the number of genes you can detect, and even when you go to more genes, it's hard because you sort of only have like so much real estate in the cell in terms of voxels that you can give up. So if you measure a gene that has a lot of copies and a gene that has a small number of copies, it gets difficult. So you're never really going to get not never, but it's hard right now certainly to get a very clean panoramic of all the genes in the genome. On the other hand, you have the capture-based technologies, but they don't have the exquisite sensitivity of the sort of hybridization of single molecule base. So I think that's one trade-off. I think maybe somebody will figure out a way to basically overcome, the quote-unquote voxel problem, which is that RNAs just tend to be closer to one another in the cell than the wavelength of light allows you to detect, right? I think if you can overcome the voxel problem, then you get the sensitivity and you get the completeness, and that's just remarkable. I think there's a lot of challenges, and my lab is working on some of these, which is just in the informatics of this, right? So how do you just so many questions. Like in the capture-based, how do you assign? When you get these high-resolution spots, how do you assign the capture RNA to the right cell? Because one spot might have RNA from multiple cells, and then the other thing is that once you've done that appropriately, how do you analyze? I mean, now you have this whole other dimension that I just don't think that you're taking account of when you analyze the data, right? So, for example, how does gene expression vary along the cerebral cortex? How does, when you look at tissue substructures and that, and then how do you really capture sort of spatio-transcriptional programs, right? Does that make sense? So.
Yeah, it does. And I think you've alluded to a couple times, folding in at the protein level, of course, is really important too, since that's where the action is. The RNA, we study RNA because it's easy to study.
Right. I mean, yeah, the miracle. Yeah, the miracle of the nucleic. I really do think that I don't know. The two things that I'm incredibly excited about to hear, to learn about, hopefully in the next 2 days, are one, RNA modifications. I think that's fascinating. I think there's a lot of biology there that we need to understand, and I think I would love to get I would love to basically have a series of technologies that puts protein analysis on the same footing as nucleic acid analysis. That would be amazing.
Some challenges for the community. Rob, thank you very much.
Thank
for
you.
your
That's my
comments.
pleasure. Thank you so much.